So in this presentation, I will be discussing about taxonomic characters. Taxonomic characters deals all those features which are of taxonomic significance. For example, if you have to identify an animal species, particularly when you have to categorize it into its different taxonomic categories, then you have to note down its so many features, its external morphology, its ecological conditions, that is habitat, its chromosome structure, several other cytological features can be considered. Its developmental stages could be considered for taxonomic purpose. So all sorts of features, presently even molecular characters are considered to identify a species. So I'm going to talk about all those features which are considered to identify an animal species. I'm going to actually explain this aspect in case of animals, particularly in insects, but it is equally applicable for other kinds of organisms also, because I'm going to give you some basic idea regarding taxonomic characters. See, here you can see, particularly for animals, these are the features which are considered for taxonomic purpose, like morphological characters. Under morphological characters, you can consider these points like general external morphology of the animals, then spatial structures, particularly genitalia. In case of insects, the male genital structures are of much significance. One can identify the structure of claspers or edigus, which is a specific structure in a particular species. Then you can also identify internal organs which come under anatomy, that is organs related to digestive system or respiratory system or excretory system, like in case of invertebrates, nephridia are there. So type of nephridia or type of kidney. Then blood vascular system, reproductive organs, internal organs. So all these come under internal morphology. You can also consider developmental biology, that is embryology, the structure of egg, type of larva or nymph, then pupal stages, their structure. And then you can also consider embryological and karyological, that is the chromosome structure, and cytological features if specific features are related to the cytological aspect of the animal, then those can also be considered. We know that every species of animal possesses a fixed number of chromosomes, and not only that, the karyotype of such chromosomes will be fixed in a particular species. So that also forms the basis of identification and could be taxonomic features for the identification of a specific species. Karyology or chromosome structures are of much importance. Then in case of animals, behavioral characters can be considered like courtship pattern, mating songs, and other isolating mechanisms. Then ecological characters are of taxonomic importance like habitats and host, food which is being consumed by those animals, because certain animals feed on a specific kind of food only, others could be omnivorous. So that could also be considered for taxonomic purpose. Seasonal variations, parasites, host reactions. Then you can consider the geographical characters, that is general biogeographic distribution patterns. We find that certain types of animals are distributed in a particular geographical region, so that could also form the basis of identification. Sympatric and allopatric relationship of populations. Then physiological characters like metabolic factors, body secretions, genic sterility factors. These come under physiological characters and also form the basis for identifying species. And finally, we shall be considering molecular characters, which are particularly seen these days, like DNA barcoding is one of the uh, popular molecular characters used for taxonomic purpose. So let us see these aspects one by one. All sorts of external features form the basis of identification for morphological characters. that is in case of invertebrate animals, we observe the external morphology of the animal, that is their body organization. Okay, we see whether they have cellular grade of organization or tissue grade of body organization or organ system grade of body organization. Then, like in case of poriferans, cellular grade of body organization is there. In case of cnidarians, tissue grade of body organization is there. And in higher forms, we find organ system grade of body organization. So that forms the basis of taxonomic identification. Then kind of symmetry, whether radial symmetry or bilateral symmetry, what kind of symmetry is there. Developmental pattern, segmentation in the animals, type of locomotory organs present in them, parapodia, and appendices, like whether the appendices are jointed or they are not jointed. Then exoskeleton, type of exoskeleton present in different parts of the body. Then respiratory organs, type of nephridia present in the animal, specificities of blood vascular system. We find in some cases the blood vascular system could be closed type, in others it could be open type, so that is also considered for classification purpose. Eye structure, shell and its organization pattern, food types, particularly in mollusk, shell and its organization is there. That is, we find that whether the animal is bivalve or univalve, or so many shells are there, and the type of food that also forms the basis of classification. Then among insects, because insects form a very large group of animals. So among insects, features like antennae, wings, thoracic and abdominal segments, mouth parts, copulatory organs, legs, etc. are considered for taxonomic purpose. And in case of vertebrate animals, there could be several features like the type of fins they have, their structure, scales, limb morphology, feathers, hairs, several other integumentary structures, nasal openings, position, barbels, internal anatomy like gills or pulmonary organs, heart structures, kidneys and their tubules, sense organs, the type of sense organs and their location, and their distribution pattern. All these form the external morphological features to identify a particular animal species. Here in this diagram, uh, you can see the mouth parts of mosquito. See, two very common species of mosquito are there. Just for understanding, I have taken this diagram so you can have idea that how identification could be done. Here the mouth parts of Anopheles, male and females are shown. Likewise, the mouth parts of Culex, male and female um, uh, individuals are shown. So if you see the mouth parts, you can identify the uh, type of species. Like in case of male, first you see here in case of male, the antennae are plumose type. Plumose means very hairy antennae are there, and this is the characteristic of a male. In case of culex also, the plumose antennae are there, but they are females, they have pilose antennae. Okay, pilose antennae means the hairs are very, very small. They are not so developed as in case of male. So this is one way to identify male and female. Then in case of Culex female, see here in case of Culex female, the maxillary palps, see here this one is maxillary palp, it is very small. So if you have seen the presence of pilose antenna, then it is a female, and if the maxillary palps are very small in size, then it will be the female of Culex, whereas if it is quite long one, then it is female of Anopheles. The other point you can consider in case of male, see here in case of Anopheles male, the maxillary palps are longer than the labium. See here it is longer than the labium, whereas in case of Culex, the length of this maxillary pulp and labium is almost equal. So this way you can see the mouth parts of these species, and you can identify their males as well as female, and also the type of species present over there. In this diagram, you can see the copulatory structure of Drosophila melanogaster. Here, this lower structure is actually phallic organ. Phallic means this is the real copulatory structure known as oedicus. Oedicus, or it is also referred as clasper. And the structure of oedicus, that is of a particular shape and size in a particular type of species. So its structure is exactly like this in case of melanogaster, but in other species of Drosophila, the oedicus structure will be totally different. And this upper structure is actually periphallic, that is around this phallic organ or copulatory organ the accessory structures are present, which is referred as periphallic organ. So the periphallic and phallic organ vary species to species. Like here in this diagram, you see uh, these left side structures are actually periphallic organs, and then these are phallic organs. So total 8 species, 1, 2, 3, 4, 5, 6, 7, 8. 8 species of Drosophila. Their copulatory organs are phallic and periphallic organs are shown here. And this portion in the middle portion means this middle portion is actually of phallic organs, where the central portion is oedicus or clasper. And you can see structural differences exist among the species. Likewise, here these 4 phallic organs, their oedicus show differences. In this diagram, you can see the sex comb. Sex comb in Drosophila, a male-specific morphological structure found in tarsal segment of foreleg. That is, first pair of leg in Drosophila, the prothoracic leg. It possesses sex comb, and that is found in case of male only. Females lack this structure. And in every species, the structure of sex comb that varies. Like in case of melanogaster, you can see here this is a single sex comb. It is present in the tarsus portion, the first subsegment of tarsus, and the number of hairs are 10 to 11. You can exactly count them. The other species is Drosophila bipectinata, where two prominent sex combs are there, and you can also count the number of hairs. These are 5 and 7, 8 hairs are here. Then 2 hairs are in the second subsegment of tarsus. Then the third species is Parabipectinata, where the number of hairs are smaller in size. Otherwise, the pattern or the type of number of sex combs are almost similar. So this is Parabipectinata. And in this case here you can see pseudoananaceae. Here the sex comb pattern is different. The last one is Miller-Cartliana. Here the number of hairs are 1 followed by 3 and then again 1 followed by 2. So this is the pattern of sex comb presence in case of Miller-Cartliana. So see 1, 2, 3, 4, 5 species of Drosophila are shown here, and they are males. They possess variation in the structure of their sex comb. So that forms the basis of identification of these 5 species. In this diagram, you can see the pattern of light flashes in 16 species of fireflies. I just told that behavioral characters could also form the basis for taxonomic characters. So you can exactly count 1, 2, 3, 4 like this, total 16 species of fireflies, and the pattern of light which they emit during night or at the time of evening, that is shown here. Here you can see in the first case the pattern of light is there, means at regular intervals light is being emitted. In the other species, little amount of light is being emitted, but the frequency is little bit more. In the third species, the frequency increases. Here, the intensity of light is very high and it is being emitted at regular intervals. So the pattern of light emission in these fireflies that varies, and it is a case found in genus Forturis. Okay, and all these species are found in North America, and the pattern of light emission during evening is shown in them, and that forms the basis of their identification. Then you can see the courtship pattern. Courtship pattern can be recorded in the form of graphs. and the courtship pattern in 4 different species of Drosophila is shown in this diagram. Here in case of Drosophila virilis, you can see the intensity of vibrating the wings at the time of courtship is shown. In case of Drosophila montana, the pattern of courtship is different. Actually, all these things show the type of wing vibration by male at the time of courtship, and that could be recorded in the form of line diagram. And this is being shown that species-wise variations are there. In case of D. littoralis, here the pattern of vibrating vibration of wings are different. Likewise, in case of Drosophila flavomontana, this pattern is totally different. So on the basis of, if you could record the courtship pattern, then you can identify the species. Here in the description, you can see it is written that 40 species of crickets in North America have been discovered on the basis of their sound analysis. A species of praying mantis can be identified on the basis of their egg cases, and the pattern of web construction by mites and caterpillars form good taxonomic features. So behavioral pattern that can also be considered for the identification of a species. I just said that karyology and cytology could be used for taxonomic purpose. Okay, so here you see in this diagram, these two are the polytene chromosome structures. This one is polytene chromosome of Drosophila melanogaster, and the other one is of a different species. So what we find that the binding pattern in these chromosomes that will vary species to species. Species to species, the binding pattern that varies. You know that in case of Drosophila, there is a chromocenter from where the different chromosome arms radiate. And those who work on this subject, they are well acquainted with the binding pattern. They can identify X chromosome and autosomes. And in certain species, you will find just 5 arms. In others, there could be 6 arms. Okay, so based on the type of karyotype, the number of chromosome arms that vary. If a chromosome is acrocentric, then almost a single chromosome arm would be seen. But if a chromosome is metacentric, then it will be represented by 2 arms. So polytene chromosomes are used for taxonomic purpose. Here you can see the metaphase diagrammatic presentation of metaphase chromosomes. In this species, you see 1, 2, 3, 4, 5, 6. 6 pairs of chromosomes are there. In the other one, only 5 pairs. 1, 2, 3, 4 pairs of chromosomes are shown in case of B. Here in this case, 1, 2, 3, 4, 5, 6. These are the pairs of chromosomes present. And here you can see the difference between male and female karyotype. So karyology and cytological features are used for taxonomic purpose. You know that every species possesses a fixed number of chromosomes. And then when you prepare the metaphase chromosomes, you will find that variation do exist. You can see the type of metacentric or submetacentric or acrocentric, telocentric chromosomes. And if you arrange them properly in proper karyological form, you can see the variations between or among these species. Then you can consider ecological characters for taxonomic purpose. And here breeding grounds of 6 sibling species of Anopheles mosquito is shown. See in this case 1, 2, 3, 4, 5 and 6. 6 different species of Anopheles are shown, like Anopheles maculipennis, Anopheles melanone, then Anopheles messi, Anopheles atroparvus. So these are 6 species of Anopheles, and the habitat, that is their breeding grounds, are shown. One is breeding in the cool running fresh water, others in rice fields fresh water. This third one is found in cool standing fresh water, rather in this water it breeds only. So in certain species breed in the brackish water. Okay, some breed in the running water, others in the standing water. So this kind of variation is there. It means the habitat that also forms the basis of identification. Likewise, Aedes species are also shown here, and the type of ecological conditions they prefer for breeding purpose, that is shown here. For taxonomic purpose, as I said, that molecular characters are also considered. People started different molecular techniques to identify variation among the individuals of same species, as well as to identify different species. And for that, immunological distance was one of the methods to identify different species. Then protein polymorphism, that varies species to species. Amino acid sequences in the protein are considered for taxonomic purpose. DNA hybridization, it also formed the basis of identifying species. Mini satellite and microsatellite variations could be used to identify different species. And restriction endonuclease analysis, that is RFLP, can be used for species identification. But presently, DNA barcoding has become much popular, and this is actually applied to identify different species, because technologically we are now so sound that we can isolate DNA, we can amplify a specific segment of mitochondrial DNA or 16 years ribosomal RNA, and that can be used for taxonomic purpose for all sorts of organisms. See here, in case of DNA barcoding is a DNA-based taxonomy tool to identify species based on the pattern of nucleotide sequence in a fragment of DNA of a particular species. The most commonly used DNA barcode region for animal and some protist is a portion of cytochrome C oxidase 1. This is of importance, that particularly the mitochondrial DNA, which encodes for cytochrome C oxidase 1, that is used, that is coenzyme 1 or COX-1 gene. It is used for DNA barcoding. The size of this gene used may be approximately 700 to 1500 base pair. This region has been widely adopted for its variability across species and its relatively conserved nature within species. So this cytochrome C oxidase 1 is a mitochondrial gene found in the mitochondrial genome of insects and in other species also, not only insects, but in other species also, in other eukaryotic species. It encodes a protein that plays a crucial role in the electron transport chain during cellular respiration. And this particular gene is highly conserved within species but shows variability between species, making it an ideal candidate for DNA barcoding. And you can go through this table. It is of much importance because these DNA segments used for barcoding in different groups of organisms is shown in this table. Like in case of animals, the segment of DNA which is of um, taxonomic purpose could be cytochrome C oxidase 1. Then it is 12 years mitochondrial ribosomal RNA gene, 16 years mitochondrial ribosomal RNA gene, and 18 years ribosomal RNA gene. These are of DNA barcoding purpose. In case of plants, MATK, that is maturase K gene, RBCL, this PSBA tRNH is used. Then ITS, that is internal transcribed spacers. These are the segments in DNA molecule which are used in case of plants. In case of protist, internal transcribed spacers. Then again coenzyme 1, then RBCL. So these are the different 18 years ribosomal RNA, 28 years ribosomal RNA genes are used for DNA barcoding. Likewise, in case of fungi, here is the name of those segments of DNA which are of taxonomic purpose. And in case of prokaryotes also, the portion of DNA which is of taxonomic use is mentioned in this table. So you might have got idea that what is taxonomic characters. That is all those features which are actually used to identify an animal or any other species of organism, and then that could be used for taxonomic purpose.